Okay, so today we're going to talk about stem cells. Specifically within this lecture, the things that we're going to be able to define out are what are stem cells, what are the different types of stem cells, why we study them, what therapies can they provide, and then some of the challenges that are faced in stem cell research is what you're going to be investigating within the module. So let's talk about what all stem cells can do. So all stem cells can self-renew and differentiate. So what does it mean to be able to self-renew and differentiate? So many of you remember maybe from biology that your cells undergo a process called mitosis, right? We have a parent cell right here, and it divides into two. And so one cell stays as the parent cell, and then we get a duplicate of that, and we call these daughter cells. And so we get two identical daughter cells when a cell goes through the process of mitosis. Well, our stem cells can do this. They can self-renew. It's what we call symmetric cell division. They do this equally, and the cell itself is not differentiated into anything specific. It's just a cell. Well, what makes our stem cell so unique is their ability to differentiate. They go through a process called asymmetric cell division. So you have your stem cell, it divides, but it doesn't make a duplicate. The stem cell remains the same in one half of the mitotic phase, but the second cell, the second daughter cell, is what we call a progenitor cell, and we can actually take that and turn it into whatever we want. So we go through the process of cell division, but the second cell actually gets designated a job, okay? So it becomes what we call asymmetric cell division because this is now a duplicate of the original. And so what makes stem cells so unique is their ability to self-renew and then maintain their own population right here through this parent line, but then also differentiate at the same time into something that we want it to be or want it to do. So there are three main types of stem cells. The first is the embryonic, which is the one that we will talk about the most in the unit because it's the most highly controversial. These are called pluripotent cells, and they can form into any cell type in the human body. The second are multipotent, and these are specifically adult stem cells that are they're limited in what they can turn into. So some types of things like blood, brain cells, and liver cells, but not every cell in the body. And then you also have what are called induced pluripotent cells. These are the genetically engineered cells that scientists work with that act like embryonic stem cells. So these are your main types. You also have the term totipotent, so it's another term that you'll see, which are the same as the standard pluripotent cells in embryonic tissues. So totipotent, pluripotent are kind of the same term. Induced pluripotent are the ones that we try to act like embryonic stem cells. So you get the idea that those terms are kind of interchangeable. Okay, so those are the types of stem cells. So now what I have for you is just kind of the idea of how conception works and how in vitro fertilization works, so that you can sort of also take that and synthesize it into what we need with what we do with stem cells. So normally, to make a baby, sperm mates the egg in our fallopian tubes and fertilization occurs. When a child can't have a baby naturally, they go through the process of IVF, or in vitro fertilization. So what that means is a fertility doctor uses hormones to make the woman produce between 10 to 30 eggs instead of the standard one egg that normally is produced. And we remove those eggs from the woman's body. The man will donate sperm, and the sperm is sucked up into a needle like you see here. And then we inject basically the sperm into the egg to try to ensure fertilization occurs. So here's some photos of the injection process here, and now we have fertilization occurring. So you start to see fertilization, so you can tell the egg is fertilized because you start to see some division happening. So this is mitosis going on in the cells. The cells right now are undifferentiated, unknown cells, and they begin to divide more and more until we get a fully fledged functioning embryo. And so here is more division continuing on. So the embryo is forming. Here goes more. So one cell becomes two, two becomes four, and so on. You know that process. And then we start to see the development of the zygote. Okay. And so what happens is on day 5, so notice this was in sequential day 1, day 2, day 3, day 4. Here we are in day 5, and day 5 we now are, what we have is called a blastocyst and a developing embryo. And so it's a big day. This is the day that fertility doctors generally will implant the embryo into the woman's uterus so that the woman can then begin to formulate the baby within the body. But it's at this stage where our cells are most specialized, and the outer layer of cells become the placenta. And there's a clump of cells, though, that sit right inside that become the embryo and later into the fetus. This is the stage where the embryonic stem cells can be isolated. Okay, and so this is what we talk about with embryonic stem cells. The process of in vitro fertilization itself isn't easy, because we have to implant a lot of these in order for them to take. Usually we can implant anywhere from between this 10 and 30 eggs, and we'll only possibly get one embryo that will develop out of that. So it's very challenging to do IVF. So you can imagine that this particular process is one of the key ways that we also try to obtain embryonic stem cells. So we do 30, 40 of these to get viable embryonic stem cells as well. And so the leftover embryos that don't end up getting used will get frozen. And so we get into this whole ethical debate of, okay, they're frozen, they're not ever going to be implanted, they're not ever going to turn into babies, so why can't we just use the leftovers that are there? So those are kinds of things that we get into when we start talking about the ethical issues involved with embryonic stem cells. So this, again, just kind of shows you the process of what happens in sort of a diagram form. So what we have here are our totipotent cells, or pluripotent embryonic stem cells that we take. And when we take them, these totipotent, pluripotent cells, and then we turn them into all of these variations of cells. And so it's at this point right here, the cell can form all of the cells in the human body. Here, this one can form almost every cell in the human body. So this is what's really super important when we talk about what is happening in terms of embryonic stem cell research, is we're taking it from something that the argument is it alive or is it not alive? And so this is just a electron microscope view of human embryonic stem cells in a culture. And here's another one which I just thought was kind of cool, which is a fluorescent image of those. So here's our current, what do we do, what diseases do, and what do we have? So currently we have blood diseases, immune diseases, things that are currently being treated with stem cells, genetic metabolism disorders, tissues, organ placement, these are just some things. And our potentials to treat are heart disease, neurological diseases, stroke, type 1 diabetes, macular degeneration, cancer, HIV, spinal cord injuries, multiple sclerosis, ALS, liver disease. There's a lot of options here to what is potentially able to be treated versus what we currently treat. And so what I have over here for you is what we do with adult stem cells. So these are the multipotent stem cells. So adult stem cells come from bone marrow. We extract them out of bone marrow, which many of you may have heard. Bone marrow extractions are very painful for the individual, but they recover from them. And in this case, what we do is we take the adult stem cells from the bone marrow, then we signal them. Basically, we take them, put them into a petri dish, and we signal them to turn into what we need them to turn into. And the key to this, though, is that the bone marrow is, of course, part of how we produce blood in the body, so it has to match the patient's exactly. So if the patient that we're trying to use the stem cells for isn't an O-positive patient, then we're not able to use necessarily their stem cells. Now, that varies. I mean, there's just, O blood type is the universal donor, and AB is universal acceptor, and things like that. But we're ignoring the sort of the blood information there, and we're just talking about how it does have to match. Otherwise, we will have some rejection. And then we put these into the patient that is needing the infused stem cells, and then they will take those stem cells. The body will take those stem cells eventually, hopefully accept them and turn them into what is required. But again, this is adult stem cell research, and so it's only going to be useful for brain cells, blood cells, and liver cells, things like that. Very, very specialized. They are powerful. They are promising. We do know that we have researchers studying embryonic stem cells, but we also know that adult stem cells are kind of more accessible to us, and we can have consent. And that's where we get into our whole idea that consent is important. We have to be able to consent to that. So why do researchers study embryonic stem cells if adult stem cells are there? And that is because, again, they're limited to what they can do. And then, of course, they're inaccessible. And then there's the idea that, we're killing an embryo to receive those stem cells. Okay. So let's go on to some of the challenges. The differentiation of the stem cells into a mature functional cell is tough. There's potential for tumor formation because uncontrolled cell growth leads to cancer, so we know that that's a problem. There's potential for immune system rejection, depending on what we're taking from and who we're taking from. So these are some of the challenges that are faced in embryonic stem cell research, and they're kind of big deals because we don't know what the outcome is. It's a lot of unknowns. Induced pluripotent stem cells is what we're working on now. This is genetically engineering new stem cells by making stem cells pluripotent. About several years ago, a Japanese scientist worked in San Francisco and made a breakthrough showing that adult skin cells can be reprogrammed using viruses like we see before. And the expression of these genes regenerates the stem cell phenotypes within the body. The virus sort of changes it and modifies them to become things that we want them to become. So there's a lot of uses for stem cells. There's a lot of things that are being done. Induced pluripotent would limit the need for embryonic stem cells. Adult stem cells are going to continue to be used because, again, we have consent and we can do a lot with them, but it's sort of a big argument as to, do we take embryos for this use? And of course, keep in mind, a lot of embryos are gotten rid of. We don't use them. They're thrown away anyway. So the argument of them being alive or dead is kind of a big deal. So there's your information on stem cells. Let me know if you have any questions.